I think when we first started studying these things, we didn't realize how important the mind-body connection was. There's just no not paying attention now to things like depression, loneliness, stress, all these kinds of what you might call mental challenges. They affect your body.
From the University of Southern California, Leonard Davis School of Gerontology, this is Lessons in Lifespan Health, a podcast about the science and scientists improving how we live and age. I'm Sean Curran, Associate Dean of Research.
And I'm Orly Bellman, Chief Communications Officer. On today's episode, how University Professor Eileen Crimmins is leveraging technology and time to understand the factors that make some people age faster or slower than others.
Professor Crimmins is a member of the National Academy of Sciences and the National Academy of Medicine. Her recent research on U.S. populations has shown that the prevalence of dementia has fallen, that both men and women over 50 reduce their risk factors for heart disease and stroke, and that Americans may be aging more slowly today than they were 2 decades ago. She sat down with Orly and provided an overview of past and current population trends and a sense of where the future is heading.
So we're very thrilled to have you participate. Can you just briefly talk about what's happened maybe over the last century and then more recently?
Okay. Well, over the last century, the U.S. gained markedly in life expectancy and practically doubled from approximately 40 to approximately 80. And the U.S. was a relative leader in that up until maybe a later part of the last century. It was noted by the 1980s. We were in the middle of the pack. We probably were at the leading edge of the pack of developed countries before that. At that point, we were in the middle of the pack. And subsequently, now, we've continued to fall behind everyone else that we would have thought we should be compared to, all the other major rich OECD countries like us. But now we're down with states that were once part of Russia, Cuba, Chile. We're below many of those states even now. So the U.S. in life expectancy has really fallen behind the people we think should be our peers. So that's a pretty serious issue. We know a significant part of it is not due to old age mortality. It's due to middle age. And it's due to, in the most recent change, has been the opioid epidemic. At the oldest ages, we still are a leader in life expectancy. So above age 74 or 5, something like that, we are one of the top countries in life expectancy. So there's quite a bit of difference between getting old and being old in this country. It's a country where we're less likely to have people get old. But once we get to the point where we are old, we do pretty well.
And what about health as we age? Can you talk a little bit about what health span is?
So in the last century, when life expectancy increased, it was because babies and children survived. When they survived infectious conditions. So they didn't have much residual from that. We sort of knew that when life expectancy increased, health also improved. When older people started being the source of gains in life expectancy, they came primarily from declines in heart disease, but it wasn't because we were preventing people from getting heart disease. People were living with treated heart disease, and people were being prevented from dying. So that was a very different set of processes that were leading to increases in life expectancy. So that's why we started to become interested in health span, to see what had happened to the health of the population at the same time that the life expectancy was increasing. And there we realized that life with disease is certainly, the length of life with disease has certainly increased. But what I would say is some things that we've done very well at putting off, we're starting, perhaps, we have a little bit of evidence, we're starting to put off disease. We think it's because we've been able to now control hypertension and cholesterol pretty well, and some other factors with treatment and drugs, basically. The prevention is mainly that more than good behavior, because perhaps smoking cessation has been a factor in that. So we're just starting to see a little bit of improvement in the age and onset of disease. And for instance, the likelihood of a heart attack at a younger age has been reduced. And we've just seen that recently. So we may be starting to turn the corner. Well, that's what we have to do, is prevent the biological aging, prevent the onset of disease, prevent the onset of disability, and then, and make those periods shorter. And that's the aim now.
So can you talk a little bit about biological age versus chronological age and your work in this area?
So we've kind of gotten to this idea that people don't all age at the same rate, meaning that people of the same age have very different health. And we understand that determining the initial factors that start this rate of aging require looking at this biology. Because, for instance, if I look at the end of this process of health change, if I look at who's alive after a heart attack, that may depend very much on how you were treated, which may depend very much on whether you had insurance and how often you saw a physician. So you can imagine that among people who actually have the most treatment, who spend the most money, they could actually survive with more diseases than the people who don't have that, which would give you a kind of adverse signal as to what's really going on in the population. And people could look sicker just because they were treated better and they lived, they didn't die. So that's when we said we have to really go back in this process and look at the biology of it. Now that when we understood that, that's when we, started a focus on cholesterol and markers of diabetes and markers of hypertension. But in some ways now we're beyond that because we're controlling those things with drugs. So now we've moved back to this biological aging where we try to get at more basic processes. And we do have an indicator of biological aging that we've developed that is a combination of markers that indicate the physiological status of multiple systems. People have better biological age if they're richer. We have changed biological age over time with the use of drugs and changing in smoking. So it's a good marker, but it's only the beginning. What we really want to do is get much more basic markers of cellular and molecular aging. So in a sense, the social science is moving to where the biology is, and we've adopted the geroscience ideas, and that there are these basic hallmarks of aging, and that we are trying now to measure those in people and develop new indicators of biological aging that include these measures. So that's in a nutshell what the next the next 10 years starting now is happening in human population studies.
That you are now incorporating all this biological data, is that new for the field?
Yes, it is new for the field. And part of that's technology. The biology wasn't there 20 years ago. People didn't have these ideas theoretically. Technically, the idea that you can gather substances to measure these things in large populations required a certain amount of technological change and ability to do things, fast FedEx shipments of blood, like things of that nature. And part of it is, I've been working on this 15 years, I would say, to get what I theoretically thought we should measure in the American population measured. And now it's just this year the data are coming available. So it's a long time from an idea to actually having data on a large population. And we've been very instrumental in pushing this across the world. So yes, I think people have bought it. Partly, we've been responsible for developing a network of people to integrate biological measurement.
And how are you doing that?
I work in the major U.S. study, which is the Health and Retirement Study. That study has people all over the country. We do not do Alaska and Hawaii, but we do the rest of the country, the continental United States. We hire a phlebotomist who goes and gets blood. We then ship it instantly to a lab, and we have a whole set of things that are done there. So that's what we do. But this has been a process. We've been collecting blood in a variety of ways since 2006. And I think at this point, we have probably encouraged about 200,000 people to be included in the collection of biological data with large aging population surveys. And that's because there are sister studies of HRS in another almost 40 countries. And those studies have sort of followed suit or come along with us at the same time. I think today we're leaders, but this is a business where it's actually good to have competition, because if some other country does something really fancy, then the U.S. will want to do it next. So it's been a very cooperative thing, and I think it's spread across all aging studies now.
You started out studying fertility and early life, and it's interesting because I've read some of your studies, and it seems as you look at aging that you're sort of back to looking at early life.
It is true. I didn't think it was going to go there, I think, when I started looking at aging, but as time has gone on, more and more people have identified early life as having a variety of pathways that affect later life.
Can you give a couple examples?
Adverse childhood experiences that might be you are mistreated by your parents, you are hungry, you are heavily stressed because you don't have a routine life, you have to move, you go with different relatives, those kinds of severe things. Your parent's an alcoholic, your parent has a mental disorder or depression, and any number of things that can add to the stress and difficulty of childhood. They're called adverse childhood experiences typically in the literature, and there's a list of things people usually ask people about. Those things appear to have an early effect on epigenetics related to inflammation, which is both sets in motion things that affect your responses to bacteria and to viruses. And then this is something that is set in motion. If it's set in motion early in life, then it's something you're constantly using to react to the world throughout life. And so that's the sort of thing we're investigating now. And we do old people. Typically, we interview people who are over 50, but more and more we are now joining with people who do midlife and who do childhood and who do actually even babies, pregnant mothers and newborns, because we really think we can sort of better trace through the life cycle at this point in time by having these groups together and comparing reactions across different people at different ages. If we wait and get a cohort of babies and follow them through their life, it's 70 or 80 years till we know the outcome. But if we make these synthetic cohorts of people at different ages now and look at the same markers and how they change with experiences, we hope to be able to understand better what's happened to old people who are today old.
And does it offer some hope for better outcomes?
We can identify all kinds of factors like that it could be treated behaviorally, environmentally. Get people out of an environment that's caustic for them, understand the costs of an environment, intervene early, and then treat. So yeah, it offers a hope. And it's more personalized than other things. So, people are into personalized medicine, but it is true that there are people who are more or less susceptible to certain things because of either their genetic makeup or their past exposures.
So I know in some of your recent studies in the last couple of years that you've identified that we can delay aging, that there's less dementia. Maybe if you could run through some of the good news.
I think there was good news about dementia, and I do believe that, there are good things and bad things. When I got into this business, it was because the mortality of older people was falling markedly. And now that fall is continued, but it's still a little slower. If I had been thinking this way 40 years before that, what I would have noticed was those same cohorts of people were the people who got quite tall. We really increased in height up until like 1980s. And that was probably a part of the good thing that came from our earlier decline in mortality. That was because we cleaned up the environment, we cleaned up the water, we cleaned up the sanitation, we developed vaccines, we developed antibiotics. So we pretty much started to live a childhood where, yes, we got some conditions and diseases, but we went and we got a shot or a pill and we were better. Or things that we got vaccines for, we just didn't get. So we were a healthier generation than our parents, than our grandparents. Those things that we were a healthier generation, I think made us become a healthier old person. We had better bodies when we got up there. And we probably had less innate damage to our organs like our heart. We had less challenge to it because we hadn't had so much infection. We hadn't had developmental issues because our mothers were healthier, when we were born. So essentially that some of that old age mortality decline was, I think, due to a young age improvement in building a body. And that a lot of people miss that in thinking about what caused the mortality decline among old. But the same people that started with that are the people who now have had a decline in dementia. So I actually really think the decline in dementia is sort of this cohort just aging through, growing taller, reducing heart disease, and now declining dementia. They had a better body. They also had a better mind. So all these things changed at the same time. We got higher income, we had less disease, we did better, we had more education, we basically were better suited. So we get better and better and better and better, and then all of a sudden we kind of plateau, and that's where we are. But so, for instance, height stopped increasing in this country, and not because of immigrants, but height of native-born people stopped increasing in the mid-'80s. So, that's the sort of the first thing that improved was the first thing to plateau. So this as a force, but other things can then take effect. So I don't think that explains the whole decline in heart disease, but I think it's a factor that was missed. There were major changes in treatment and surgery and major things like that too, but there was a mixture of factors and they might have been working on better hearts. The same thing now, we might have better brains, that might be the first thing, and eventually, hopefully, we get better drugs. Today there's a talk of a vaccine that maybe will work. I think it's been tried on 46 people so far.
What else is good news?
Well, we better understand health. We do know how to treat lots of things. We are able, I believe, for people who have a disease to extend their life with the disease. That is good. We probably also extend the time from the time you get a disease to the time you could become disabled by the disease. So that's good. You still have it, but you're functioning. So that's the point. So yes, there are things that are good, but sometimes the more we do that's good, it tends to look bad. So the more mortality declines, the more sick people are in the population, because you save the sick people from dying. And the population looks sicker, but it came from a good thing, which was really what you spent your healthcare money on. So that's why it's this kind of complex interplay of things, that the good things can make things look bad.
Right, because if you're not sick, you might have been dead.
Right, because that's the alternative. Yes, there are good things. The bad thing, I mean, the social differences, the fact that we have large parts of this country where mortality has not declined for over 30 years. The fact that middle, white, low-educated women look terrible. They have probably taken the brunt of some social changes that made divorce more common, that left them without jobs and without families to support them, but with children that were their job to support. So there are things that are problematic. There are parts of the country is dividing more into two parts. There's the coasts and some of the upper part of the country that tend to do pretty well. And then there's this band through the bottom, red states in many ways that have poor health. They also have bad policies. They did not become a part of the group that that covered many of its people with health insurance in recent years when it became possible. So they had worse health and worse health insurance, and they have worse economy. So you get a lot of bad things are coalescing in one place. And it's also true that the bad things are coalescing in people. So in the old days, the first people to smoke were well-off people, were educated people. Well, the last people that are still smoking are the poorer people and the low-educated people. The people who are doing drugs are lower educated and poor. The people who are over-drinking are lower educated and poor. The people who have the worst diets are lower educated and poor. So you have all the bad things, risks, coalescing in people that are worse off than economically, socially, physically, and they live geographically in certain places. So it's a country that once was much more equal across the country, in many ways, pulling into two. For some people, progress just continues, and that's people with high education and good jobs, professional jobs, people that are part of the modern economy and who don't do those risky behaviors and who pay attention to diet and exercise. And it's a world that has become bifurcated in many ways.
And you mentioned low-income white women. Are there other groups that are...
Well, there's always, Black population has always done worse. They've always had a shortfall in life expectancy. It has actually diminished. In the difference between whites and Blacks has been quite cut in the last few years, which that's a very good thing. It's been cut by about half. And in fact, the group that has improved the most was Black males, who were in the worst state. They had the most improvement. So that's a good thing.
And another group that is more equal now, is it in heart health, men and women, or in cardiovascular risk? Is that right?
Yeah, we have shown that the pattern has changed markedly. We used to think men had much worse cardiovascular risk factor profile from women. It now looks, from what we see in the U.S., and we use large national data, that men and women have a much closer to parity in their cardiovascular risk factors. And that's because we control many of those with drugs, and the control now is pretty equal for men and women. It's initially, when we first started controlling hypertension and cholesterol, men were somewhat more likely to get that treatment. Now it's if you have risk, the likelihood is similar for men and women to get treatment. And what it means is it's not just, if we look at a number of risk factors for men and women at a given age from say 40 through 80, they look the same, but it's also true that the age differences have gone away. And that's pretty surprising. So I sort of say that's why we're beyond cholesterol and hypertension now, because it's so controlled that it's hard to know what the real physiological state of people is underneath, because so many people are on drugs that if it doesn't differ by age, that's a good thing. It means it's not going up, but it also means that we need to look at other things that still do differ by age.
What are your future research directions, or where do you see things going?
Well, my future research directions are in extending this understanding of the biology that changes with age, that differs for men and women, that causing them different outcomes, and that differs by social and economic status. And so I'm trying to move to this more cellular and molecular level to understand how much of that, the differences that we continue to observe, can be explained by these biological changes that may differ in their timing of change among these groups.
Is it possible to give an example? What would be a cell or tissue you might study?
Telomere length is one. So for a while, people touted that as a marker, an overall marker of cellular aging. And then the results have been very up and down and mixed, and people have sort of not as interested in that marker anymore, but we have actually developed a subgroup to work on this because we think it's been badly measured. And so we want to get the truth about that. And why are blacks and whites different in a way that you would not expect? So that's one. Methylation, that is a change that occurs to your DNA with exposure to something bad or good. You know, some kind of exposure changes this methylation level at different points. We want to understand what are the markers that indicate something has gone wrong. And we have a lot of other RNA-based markers that we're trying to develop now. We're focusing heavily on inflammation, and just recently we've developed a data set that has, at a cell level, we have what kind of T cells and B cells, you know, a national sample of people have by age. And that's never been done before for a human population. Only for large, I mean, if you put all the studies together, they might have 1,000 people. Well, we have 4,000 people. And we have this extensive set of what your cell subsets look like. So that's what we're looking for. You know, have you used up all your naive cells? Do you have nothing left to fight off the things that happen to you in old age? You know, is that why people are getting cancer or succumbing to pneumonia? If you try to look at the health of the immune system, we can tell that people who are poorer have a worse immune system. Now, it may be they were exposed to infections earlier. One of the things we look at is CMV, which we basically all have been exposed to this virus, and have it in our bloodstreams by the time we're 80 or 90, but maybe some people had it earlier and some people have other things that make it get out of whack, and then it takes the rest of the immune system down. This is what we're looking at.
So you're looking at a lot of different
markers
and
and we're trying to figure out how to put them together to get a whole person's biological age.
And which one of them might...
Which one is controlling the set of markers? We're looking at components and we want to put it all together. That's sort of what we're doing in the next 10 years, probably.
That's really interesting. I wanted to ask one last thing about, you're talking about serious biology here, but then there also seems to be this realization that there's also either environmental or mental health, that is a big part, how lonely someone feels.
Yeah. I think when we first started studying these things, we didn't realize how important the mind-body connection was. There's just no not paying attention now to things like depression, loneliness, stress, all these kinds of what you might call mental challenges. They affect your body, and the mind-body connection is quite strong. So the signals from adverse mental psychological states are strong to certain parts of the physiology of a aging person.
Is there anything else you want to add? Any other things that have surprised you over the course of your studies, or things you think are important as we?
Every year it's something new. I never would have predicted I was doing this kind of thing. I wasn't trained to do biology. I took data as they came. I didn't think I'd spend my life developing data. But now, there's just something new all the time. I guess I'm done being surprised, because every day there's something new. And every day every year, I'd say we're dealing with a different set of ideas on these things. So.
And do you ever take any of the things you learn from your research or elsewhere and try to apply them to your life?
Probably not, because if I did, I probably would relax. I probably would take weekends and nights off. There's lots of things I've learned that I ignore. And I probably wouldn't eat as much chocolate or butter. Both of which I think make me pleased with life.
Which is important.
Yes.
That wraps up this lesson in Lifespan Health. We'd like to thank Professor Eileen Crimmins for her time and expertise, and all of you for choosing to listen. Tune in next time for another lesson in Lifespan Health, and please subscribe to our podcast and learn more about us at lifespanhealth.usc.edu.